Hi, this is Thomas Bjorkman. I'll be talking to you today about a piece of research that's part of the Eastern Broccoli Project. The Eastern Broccoli Project is a NIFA-funded project intended to develop broccoli adapted to the East Coast and a concomitant industry here. To do that, we need to increase the genetic diversity in the gene pool to allow adaptation to the high humidity and warm summer nights characteristic of this region, but not to any of the current growing areas. This is part of the PhD work of Zach Stansell, and we expect that it will be published as part of a larger work soon. So broccoli in the form of landraces has probably been around the northern Mediterranean for thousands of years as a popular winter vegetable. But the broccoli that we know from the stores today has a much more recent origin. The breeding for it only began at the turn of the 20th century, and hybrid broccoli development for the international standards only began in the 1960s. But many international seed companies have been breeding for broccoli recently to meet the very uniform market requirements on the international market today. Now that uniformity that we see could be the result of a large reduction in the genetic diversity relative to the land races. But on the other hand, they are also trying to bring in traits of disease resistance and adaptation that could come from other sources, and that could increase genetic diversity over the land races if they're going beyond the original broccoli germplasm. So the question we were interested in is whether this uniform shape that we see today means that there's less genetic diversity. In order to ask this question effectively, we needed a collection of germplasm that accurately reflected the genetic diversity that exists in both the land races and the modern F1 hybrids. So we were able to get germplasm from various collections that had been made in previous decades, and also germplasm that represents the most recent breeding developments from active breeding programs throughout the world. If you look at the broccoli in the picture of the 109 accessions that we had, at this resolution you can see that they look generally like broccoli. So we took this collection and used the technique called genotyping by sequencing to develop a high-resolution map of the broccoli and able to test genetic differences at over 30,000 markers. The high marker density gives us great sensitivity for finding variation that's explained by one gene or a few closely linked genes. Previous lower density methods can't detect where selection has acted on only a small region of the genome. The high quality of the markers also means that the differences reveal true genetic relationships. We grouped these accessions by their genetic relationship using the marker data. And we use principal component analysis, and with over 30,000 data points per accession and 109 accessions, you get really robust principal components. We ended up using 3 of them. I'll only show the first 2 in this talk. And 4 distinct groups emerged from this. The land races separated into 3 different groups, and the modern hybrids all ended up in 1. The principal components analysis separated these clearly into the 4 groups. And I will talk about the groups individually in a moment, but here they are by different colors, and we're calling these violet cauliflower, sprouting broccoli, the landrace calabrese, which is the Italian name for the regular broccoli appearance, and then the F1 modern hybrid calabrese type. We're interested in asking questions about the progenitors of the Calabrese type, such as could it be a cross between the violet cauliflower and the sprouting broccoli. The principal component analysis essentially takes the over 30,000 data we have for each accession and looks at the relationships among them to reduce that to a much smaller number, in this case 3, or what I'm showing you here, 2. So the first two principal components capture about 2/3 of the total genetic variation, about 43% in the first and 20% in the second principal component. And these are done on the genotypes, not phenotypes. So first off is our F1 hybrid Calabrese, the modern international broccoli. It has a familiar shape. Some of the features that have been bred to high uniformity include a dense head that has little airspace inside. The branches are really tight, the flower buds are small, the stem is heavy, they get paid by weight after all, there are no leaves in the head, and the side shoots are really tight so that they can handle being transported. In the principal components analysis, they group very tightly together. They're right next to the landraces, which are in orange, but there is no overlap. In principal component 2, they actually fall intermediate between the sprouting broccoli and the violet cauliflower, but on principal component 1, they match up almost exactly with the landraces. The landrace Calabrese are open-pollinated types that look like modern broccoli, essentially with one large central head, and they are actually grown all over Italy. Calabrese means that it comes from Calabria, but they are not at all restricted to Calabria. The landrace types vary quite a bit, but they tend to have much looser heads, they tend to have smaller heads, varying diameter, generous side branching, and highly variable bud size. In the principal components analysis, they end up separating cleanly from the modern F1s, similar in principal component 1, similar to the sprouting broccolis in principal component 2. Clearly a distinct group. The sprouting broccolis have many side shoots, that's why they're called sprouting. They tend to have leaves mixed in with the heads. That's a desirable part of them as a food. They have large flower buds. Now, the sprouting broccoli is most likely a previously ignored source of diversity, and they might have many unique characteristics that could be exploited in breeding and improvement efforts. There are some of them that on principal component 2 overlap decently with the F1 hybrids. So violet cauliflower is kind of a weirdo that not many people know of. And when you look at the picture, you'll see first of all it's not violet. And if you look closely, you can see it's also not cauliflower. Those are floral buds. Those are not reproductive primordia as they are in cauliflower. So this is actually a broccoli. Now violet cauliflower has the head structure of modern hybrids. Is that where they got it? In the principal components analysis, they are really quite the outlier. And looking at principal component 3 and looking also at the phylogeny reconstruction that we were able to do, it really doesn't look like violet cauliflower is the origin of the tight head in modern broccoli varieties, that it's developed on an independent track. Here's a closer look at the modern hybrids group. That's about half of the accessions. Next to the image of each one, you'll see a line, mostly green in this case. That line shows how much of the genome of that accession is composed of each of the original groups. So you can see that they are primarily the F1 Calabrese group, but there's a little bit of the orange landrace in some of them and a little bit of the violet cauliflower in a couple. Among the landraces, we find that the sprouting types come out as being almost pure sprouting type, the pink ones. Then in the second row, we have some purple cauliflowers, violet cauliflowers, some of which are actually violet, but there tends to be little intermixing there. And then finally, the landrace types in orange, we see many that are pure landrace, but then some that are mixtures. And the ones that are mixtures tend to be some of the early types, Ramoso Calabrese landraces, some that are currently on the market as Calabrese, De Chico, even Waltham 29 shows some of the landrace parentage. Here we have a map of the full genome of broccoli, and there is a lot of information in this. For now, we are only going to look at the orange-brown area so the position on the genes. There's 9 chromosomes. The x-axis is the position on the genes. That brown area is the nucleotide diversity of the F1 hybrids divided by the nucleotide diversity of the landraces. When the orange goes up, that means there's less genetic diversity in the F1 hybrids. And when the line goes down and it's actually colored in green, that means there's more diversity in the F1 hybrids. So across the whole genome, the hybrids have retained about 55% or half of the nucleotide diversity that's present in the landraces. Now to take a look at some of the interesting things going on here. First of all, we look at the genetic uniformity, where there's very little genetic variation in the F1 hybrids. We found 13 locations that account for 2% of the genome where there is exceptionally low diversity. These are likely to be places where there are essential commercial traits and the variation present in the landraces is undesirable. It's important to retain these in any wide crosses made to landrace broccolis to bring it back to commercial quality. At the other end of the spectrum, we find 8 small regions that have higher diversity among the F1 hybrids. Those 8 sections represent about 3% of the genome, coded here in green. Those are likely to be where there's been introgression into the F1 hybrid population from outside of the broader broccoli world and going elsewhere in Brassica oleracea. Finally, we found a domestication hotspot at the end of chromosome 9 through this and other work. So we're curious what constitutes domestication, and you can see some interesting things here where it's been blown up, that we have a very strong signal of increased genetic diversity at about 45 megabases, and some very strong selective sweeps going on that's taken a lot of variation out at around 39 megabases. Thus we see opposing effects on genetic diversity happening in narrow regions close to each other in the genome that has been part of the domestication process. So to conclude, we found that the broccoli hybrids in modern commercial production retain about half the original genetic diversity of the landraces that they're derived from. We think that there are valuable traits that may yet be found in the landraces, particularly ones that can help select for varieties that will grow in the east. The genetic variation can more easily be returned to broccoli breeding programs by using genomic techniques like this one. For instance, small genomic regions that are essential for commercial types can be identified and selected for through marker-assisted selection. A backcrossing program that would keep only the commercial alleles at the 13 small, low-diversity regions is an example. We also suspect that there may be valuable things to be gotten from both the violet cauliflower and the sprouting broccoli types that has not been exploited to date.